Okay, guys, we left off talking in class on Tuesday, chapter 21, about antibodies. Antibodies, remember, are produced by B cells, specifically when B cells differentiate into plasma cells. And we find antibodies in our body's humors or fluids that float around in the body, things like blood plasma, or fluid that we're going to find in tissues. We call this interstitial fluid. And antibodies have very specific makeup. What you see in the diagram here, which you can find on page 781, figure 21.14, is the basic structure or monomer of an antibody. All antibodies are made up of these structures or multiples of these structures. For an antibody is an immunoglobulin. And this is going to consist of very specific protein parts. So if you look at the diagram, you see at the top of the diagram something called the heavy chain. All antibody monomers have two heavy chain regions. There's a variable region and a constant region in all heavy chain regions of all antibody monomers. The other thing we're going to see is a light chain region. And this is kind of where the antibody looks like a Y at the top. The light chain region consists of, again, a variable region and a constant region. And then holding together not only the light chain and heavy chain, but another portion of the antibody monomer, which is called the stem region, is a group of bonds called disulfide bonds. All antibodies have the same basic structure. The difference between one antibody and the next, and this is what makes or gives the antibody its specificity, is the variable regions in the heavy and light chain. So in your diagram, at the top of the Y, you see a little purple region and a little blue region. Again, these are the variable regions that we see in each of the heavy and light chains. So this is what makes the antibody specific for the antigen that it was made for. The other very important thing to remember about the antibody monomer is some of the different things we find on the antibody monomer. The antibody monomer has places on it that are going to help bind MHC, and the antibody monomer also has places on it that will help initiate complement. One of the things you have to know is the different classes of antibodies. When you see the discussion, which begins on page 781 about antibody classes, basically what we're talking about is some of the different configurations of antibodies, those little Ys. And you see them throughout this whole chapter. There are 5 different types of immunoglobulin. And remember MAG, M, A, D, and G. These antibodies or immunoglobulins are called IG for short. And the 5 different IGs that we find in our circulatory system and in our bodies is IgM, IgA, IgD, IgG, and IgE. Now all of these have different targets and different functions. All of them are going to form a complex called an antigen-antibody complex when they come in contact with their target. This diagram on page 782 in your textbook talks a little bit about how antibodies help to neutralize antigens. When we look at the antigen-antibody complex, when an antigen comes in contact with an antibody, it can form something that's going to neutralize or mask some of the dangerous parts of the antigen it comes in contact with. So you see at the top of the diagram where it says neutralization, we see it sort of surrounds things and masks them. So they are neutralized and other cells can come and get rid of them. They also cause clumping or agglutination. You saw this in lab when we did blood typing. So when we added antibody to cells and the antigen was present, it actually caused big clumps to occur. Another thing that can happen when this clumping occurs is something called precipitation. And this is where we actually take insoluble antigens, or antibodies I should say, and create soluble particles that will come out of solution. The other thing that antigen-antibody complexes do is activate the complement cascade. So the three I just mentioned are inactivating antigens. Fixing an activation of complement is going to help get that complement cascade going and form that complex called the MAC, M-A-C, and form a MAC attack. If you follow down in this diagram, you see complement leading to cell lysis. When complement fixes and pokes holes in cells like bacteria, it causes the cells to explode and cell lysis to occur. Some of the other things in this diagram, like agglutination and precipitation and neutralization, can also be a call to phagocytes and enhance phagocytosis. So things like this can actually be part of the whole opsonization process that causes phagocytes to recognize, engulf, and destroy antigen-antibody complexes. Complement fixation can also enhance phagocytosis as well, and can also induce inflammation. Remember, inflammation is going to call cells to the area, call phagocytic cells to the area with their chemical message or chemotaxis, and can actually enhance the release of histamines, which will call more cells to the area. So the inflammatory response is going to help get cells to the area that need to be there, and that will allow the removal of any foreign substance. So antigen-antibody complexes are basically neutralizing and holding on to and calling to the area cells that are going to get rid of foreign invaders. Antibodies don't kill the antigens, they just hold on to them so other cells can do the job. Table 21.4 on page 783 gives you a rundown of the different classes of immunoglobulins. Remember, that's a fancy way of saying antibodies. IgM is called a pentamer. The reason it's called a pentamer is because it's made up of 5 separate monomer units, like we described before in the previous diagram. What I want you to know for each of these different immunoglobulin classes is I want you to know them by name, IgM. I want you to know its makeup, its structure. IgM is a pentamer. And then I want you to know a little bit about the antibody and when we would find it. For example, one of the major things to remember about IgM is when we start that first response, IgM is typically the first type of antibody that's produced and released by plasma cells. And again, this is during a primary response. It's important because it fixes complement. The other very important thing to remember is that it's going to be circulating in blood plasma, and it is a very potent agglutinin, causes agglutination to happen, clumping to happen. Think about how many different arms I can attach antigen to. In an IgM antibody, I have 10 different places that can attach to an antigen in this particular antibody molecule. The next one's IgA. We call this a dimer because there's 2 di monomer units that make it up. Remember where to find it. We're going to find it attached to epithelial cells, surface, mucous membranes, epidermis. These are kind of the first line of defense antibodies. We find them up in the nose. IgD, also found on B cell surfaces. This is what we're going to bump into to activate B cells during an immune response. IgD is called a monomer because there's only one of those antibody units. IgG, this guy is the most abundant type of antibody found in circulation. 75 to 85% of circulating antibodies are IgG antibodies. So this guy has a lot of work to do in the antibody world. He's going to be the primary response antibody. It's going to help fix and activate complement. It's going to protect against bacteria and viruses. This also crosses the placenta and is going to help with what we call passive immunity from mother to developing fetus. IgG, most abundant antibody in plasma, and it's a monomer. And then last but not least, we have IgE. And we find this on a lot of mast cells in connective tissue, on basophils. It acts as a trigger, secreted by plasma cells in the skin, mucous membranes. We also find traces of IgE in plasma as well, and levels typically rise of this particular antibody type during some sort of allergic reaction. Also chronic parasitic infection in the gastrointestinal tract. The next thing the book talks about, after it describes all of the different antibody classes and complement fixation, is monoclonal antibodies as clinical research tools. Way back when I used to do immunology and oncology research, one of my jobs was to make monoclonal antibodies. And basically what that job involved was eliciting an immune response in an animal. I typically worked with mice. And we wanted to make antibodies, pure antibodies, to very specific antigens. The reason we did this is because those antibodies could be harvested and used in different kits. For example, when you go to the doctors and you suspect you might have strep throat, a kit using antibodies, pure antibodies made from a single cell or a monoclonal antibody, can detect very specific antigens. In a strep kit, it would be looking for very specific antigens on the surface of the strep bacteria to determine whether or not it was present in the culture. So monoclonal antibodies are made so that we can have pure antibody preps that come from a single cell. We create them in the lab by fusing tumor cells, which are immortal cells, with cells of interest, those B cells or B lymphocytes that are making the antibodies we want. We create a cell, a hybrid cell, called a hybridoma. And then we have basically clone that cell, allow it to reproduce itself many, many times. In the liquid around the cell, where the cell grows, we're going to find lots and lots of pure antibodies that we're interested in, and those can be harvested and used in kits. So many of the kits, the rapid detection kits used in clinical settings, use monoclonal antibodies. And these antibodies, again, are produced from something called a hybridoma. The next part of the book talks about summary of antibody action, basically talking about the diagram we looked at previously and talking about some of the different ways antibodies neutralize antigens. This diagram ties together two of the big players in cellular immune responses. We've already talked a little bit about B cell activation, but the other cells involved in an all-out immune response are T cells. And there's two different types of T cells that are described in an all-out immune response. They are categorized based on the proteins on the surface of the cells. We talked a little bit about this last class, and we mentioned two different very specific cellular receptors found on these T cells. One is called CD4, and one is called CD8. CD8 and CD4 cells have very specific and different jobs in an immune response. CD4 positive T cells, meaning CD4 polypeptides on the surface, or glycoproteins on the surface of cells, have a very specific name in an immune response. And we call these guys helper T cells, or T sub H. And they're going to help organize the immune response. These are the guys we talked about with respect to HIV. They're going to help activate B cells. They're going to help activate other T cells. They're going to help activate macrophages. And they're going to direct the whole adaptive immune response. Also on the surface of these cells, we're going to find MHC class II, and they can also act as antigen-presenting cells. Now, antigen-presenting cells that have MHC class II and show the specific antigen to the helper T cell are what is going to activate the helper T cell. So remember my little analogy with Auntie Jen and her head. Dendritic cells that have MHC class II proteins displayed on their surface can help activate helper T cells. So once they've been activated, they're going to kind of do the same thing as the B cells did that we discussed earlier. They're going to start to proliferate. Some of them are going to make memory cells, and some of them are going to go off and help direct adaptive immunity. So memory cells are created of these CD4 positive T cells. The other type of T cell that is going to help in an adaptive defense is a T cell that has a very specific receptor or glycoprotein on it called CD8. We call these guys CD8 positive T cells. The CD8 positive T cell has a little bit of a different job. In order to be activated, the CD8 positive T cell has to see antigen presented on the dendritic cells by a different type of protein called MHC class I. Again, MHC is major histocompatibility antigens, and we find them on antigen-presenting cells, helping to show everybody in an immune response what they need to do. So MHC class I is what is going to help activate CD8 positive T cells. Now these guys are going to proliferate when activated. Some of them are going to go into memory, and some of them are going to go off and become what we call a cytotoxic T cell. This is the guy that has a little spear that can poke holes in things. These groups of effector cells are what's going to go off and actually actively fight during an immune response. Remember, this is adaptive defense and very specific. The only way I'm going to activate these cells is to show them a very specific antigenic determinant, just like antigen's head. So MHC proteins and antigen presenting are very specific. We see different types of antigens and MHC proteins on different types of cells. Table 21.5 shows us some of the different types of MHC, class 1 and class 2, and where I find them and what their job is. So remember, class 2 MHC is going to present and activate CD4 positive T cells. Class 1 is going to activate CD8 positive T cells. MHC class 1 is found on pretty much all body cells except for red blood cells. It's extremely prevalent throughout the body, and a protein fragment of 8 or 9 amino acids long. So it's not very big. MHC class 2, less widespread, typically found on the surface of cells that present antigens to CD4 positive T cells, places like dendritic cells and macrophages and B cells, for example. What's next in your book? So, don't forget to check out figure 21.16 on page 785. And don't forget to look over table 21.5 on page 786. So, activation and differentiation of T cells talks about some of the different ways that T cells become activated. This diagram, I think it's 21.17. Yes, it is. 21.17 on page 787 helps to show us some of the different ways to activate T cells. In this particular portion of the diagram, we have MHC class 2, which is basically an antigenic determinant presented by dendritic cells. And we're going to see that, sorry about that. We're going to see that presented to CD4 positive T cells to help activate those cells. So antigen presenting cells are going to basically engulf a bacteria or foreign particle, break it down, remove the antigenic determinant, and actually hold it out on its surface, just like my analogy of chopping antigen's head off, putting it on a stick and holding it out to the rest of the cells. This is going to help dock with T cell receptor proteins on the surface of CD4 positive cells, and that's going to activate the cells to proliferate and differentiate. So to start making more of themselves and then change into a cell that's going to go help with the immune response. This next portion of the diagram shows us helper T cell help in a humoral response. So again, T cells are going to sort of be the cruise directors of the whole response. Helper T cells are going to bind to different complexes on other cells and activate them and inform them of what they have to do. Please notice that antigenic determinant is always present. So it's their way of letting everybody know what they're looking for and activating them into action. So MHC class 2 proteins are going to help sort of dock cells and inform them of what they need to do. CD4 proteins are very important, and helper T cells are very important in the co-stimulation or stimulation of everybody else in the immune response. So they're very important cells in adaptive immunity. Cytotoxic T cells also have a very important role because they're actually going to go actively kill off foreign invaders. So in this diagram, 21.19, we actually see a cytotoxic T cell killing off a foreign cell. In this case, it happens to be a cancerous cell. At any given time, you are constantly making cells that just quite aren't right. So you have cancer cells in you that are being formed as we speak. The reason you don't all have cancer is because you constantly have these guys surveilling the area. And one of the things that cytotoxic T cells can do is actually destroy cells by doing sort of the same thing complement does. It forms a little complex that pokes holes in things. I kind of called it the little spear that pokes holes in things, but that little spear is actually a group of proteins that form something called a perforin. And a perforin is very similar to a membrane attack complex, and it actually pokes holes in things. Not only does it poke holes in things, but allows the entry of something called a granzyme. And a granzyme is going to actually trigger the cell to go into apoptosis, or programmed cell death. So cytotoxic T cells can poke holes in things, and then they can inject granzymes, which will induce apoptosis. Some of the different groups of chemicals important in this whole immune response are listed here in table 21.6 and described starting on page 787. We talked a little bit about interferons before. Interferons were going to help block cells from becoming infected with virus. Interferons and another group of chemicals called interleukins are all different types of chemicals called cytokines. So in this diagram you see a list of all the different types of interferon. We have alpha, we have beta, and we have gamma. And interleukins, which are a series of chemicals that are going to help with an immune response as well. We have interleukin 1, 2, 4, 5, 10, 12, and 17. You don't have to memorize these, but you do have to recognize that these are important chemicals that are going to help in adaptive immunity. We also have different chemicals called suppressor factors, transforming growth factors, and another group of chemicals called tumor necrosis factors. All of these, again, are very important in activating cells of adaptive immunity. So we have to understand the role of the helper T cell, and we have to understand the role of the cytotoxic T cell, and we have to understand the role of the B cell. So another type of cell that we also is described for you on page 789 is something called a regulatory T cell. Now, regulatory T cells are going to release cytokines, and they're going to help with dampening immune response. Because at some point, once the invader has been neutralized, we have to slow down all of the cell proliferation and differentiation and just rely on the memory cells that we've created for another attack if possible. So 790, know that little diagram. Generally be able to recognize terms like interferon, interleukin, and understand their role in an immune response. Next thing they talk about is organ transplants and prevention of rejection. Because of all the different proteins on the surface of cells, and because of the fact that your immune system is highly aware of something that is not self, when I try to transplant an organ from one organism to another, I have to take into account all of these different receptors and proteins on the surface of cells. Because remember, this is how your immune system is able to recognize something as foreign and destroy it. So a couple of terms you should be able to recognize: autograft, isograph, allograft, and xenograft. So if I want to transplant an organ from one place to another, the best scenario for me is something called an autograft. So for example, if I need bypass surgery, the best donor would be myself. So if I could get blood vessels, say from leg, and bypass blood vessels in coronary circulation, I can ensure that I'm not going to have any type of rejection. I can ensure that I'm not going to have any type of immune response from my immune system recognizing something as foreign, because they're my own cells. The best one, excuse me. The next best is something called an isograph. So if you happen to have an identical twin handy with extra parts, that would be the next best choice, because identical twins have the exact same genetic makeup. So that ensures that you have pretty much the same receptors on the surface of your cells, or antigens on the surface of your cells. Next best thing is same species. But again, we have to be very careful about making sure that those surface markers are as close to yours as possible. So when I say donate something tissue to another donor, testing has to be done on all of the different antigens on the surface of cells, and that's what tissue typing is. So we're going to look at types of MHCs, and we're going to look at all the CD4s and the CD8s, and we're going to look at all of the different antigens on the surface of your cell. If I'm type A blood, I have to make sure my recipient is type A also. So all of those antigens have to be looked at. Same species would be the next best choice. And there are actually xenograft. You can, from similar species, receive transplants. Other species that are common are baboon in heart of human. Pig, for example, is common for mitral valve. So transplantation has to take all of this into consideration so that the immune system doesn't attack and kill off those cells. This is a summary of everything in adaptive immunity. So this is one of those crazy diagrams again, but all of the players in an immune response are on this diagram. So it's a simplified summary of the primary immune response, that first time you come in contact with an antigen and what happens to your cells in the immune response. So look this over. We have innate immunity, that non-specific stuff, and adaptive immunity working together to create this immune response. So if we get past innate immunity, innate defenses, surface barriers, and some of those beginning cells that recognize foreign and get rid of, and we kick into adaptive immunity, we see in this diagram the CD8 and CD4 positive T cells, and we also see in this diagram how they are involved in activating B cells and B cells proliferating to make memory cells and plasma cells. So this is sort of a summary of specific immunity. The other thing that's going to be important in this chapter that's going to bring that information down to small is on page 792, where we see table 21.7. So there's a list here of all the different types of cells and what their function is in an immune response. There's also a list of some of the different molecules in adaptive immunity. And again, adaptive immunity is going to be more specific. Innate, some of these guys were also categorized there as well. Complement and cytokines can be very specific when they are activated in the innate end, or excuse me, in an adaptive end. But they can also be part of the innate defenses as well. So we see them here listed on page 792, and we see their function in an immune response. So have a general understanding of that as well. Some of the homeostatic imbalances of the immune system are immunodeficiencies. So problems with either the cells learning what they're supposed to learn, antibodies not being able to be made properly, and this can result in problems with an immune response. They define something called severe combined immunodeficiency syndrome, or SCID. And this could be an actual genetic issue. Deficits of B and T cells. And again, you see the importance of the role of B and T cells in an immune response. They also talk a little bit about HIV, which we discussed in class on Tuesday. Human immunodeficiency virus. And that virus attacks CD4 positive T cells. And we understand the importance of CD4 positive T cells in an immune response. The cruise director, as I call it, of the immune response, when it becomes inactive, it's hard for everybody to know what they're supposed to do in an in adaptive immunity and adaptive defenses, specific defenses. So people with full-blown AIDS, acquired immune deficiency syndrome, have trouble with their immune responses to very simple antigens in the environment, and may succumb to things that a person with a normal reacting immune system wouldn't. We didn't recognize this until about the 80s. So since 1981, there's been global awareness of how prevalent HIV and AIDS epidemic is. They talk a little bit, interesting information about treatment and prevention, and some of the different chemicals that are being used to try to slow down the progression of the HIV virus. Another thing they describe on page 794 is autoimmune diseases. And again, I refer to flunking kindergarten for my T cells, but if cells can't recognize their own cells and self-antigens, they might start attacking self. Some of the autoimmune diseases you might have heard about are described under autoimmune diseases on page 794. Things like rheumatoid arthritis, which attacks joints, multiple sclerosis, which is what we call a demyelinating disease. It actually attacks that myelin sheath. Graves' disease, thyroid issue, type 1 diabetes, an issue with insulin and insulin-producing cells in the pancreas. Again, those cells are called beta cells, and we talked a little bit about that in the endocrine system chapter. Systemic lupus erythematosus is also a problem. It affects kidneys, heart, lungs, skin, and glomerular nephritis is a problem with kidneys and kidney filtration. So this is where your immune system is actually attacking its own cells. By blocking some of the various chemicals that are being secreted to initiate proliferation and differentiation, like various different cytokines, we can kind of keep these diseases in check. So they talk about that under treatment of autoimmune disease. Failure to self-tolerate, again, is a problem with that education of the T cell in the thymus. And then they talk a little bit about hypersensitivities, when the immune system damages tissue as it's fighting off a perceived threat. Things like pollen, animal dander can actually cause hypersensitivities to occur. And inflammatory response and hive formation are some of the different things we see in immediate hypersensitivities. Hypersensitivities are described on page 795 in your textbook. Many of you have heard of allergies, and the antigens in question with respect to allergies are called allergens. So pollen can act as an allergen, and animal dander can act as an allergen. Dust mite, refuse can act as an allergen. So we see the allergic reaction or hypersensitive reaction described in this figure, which is 21.21 on page 795. So plasma cells are going to start producing large amounts of IgE type antibodies against the allergen. So when we talked about the different classes of immunoglobulins, one of the things we saw was inflammatory response and hypersensitivity to parasite. This is my IgE antibodies to allergens as well. Attaches to mast cells, and that's going to subsequently release histamines. Histamines are chemical messages that are going to cause inflammation. So we're going to see inflammation due to outpouring of fluid from capillary beds, because it's going to cause the dilation of capillary beds throughout the circulatory system. We're going to start releasing mucus as a result, and this might even cause the constriction of bronchioles. So you might see someone undergoing a severe allergic reaction have trouble breathing because of all the fluid buildup and the constriction of bronchioles. So please just have a general understanding of hypersensitivity, delayed hypersensitivity, and what an allergen is. I think that, my friends, is the end of chapter 21. Again, I'm going to post this just in case we don't have class on Thursday. Therefore, no matter what, we will have our exam on chapters 18, 19, 20, and 21 on Tuesday of next week. I have already sent emails out to everyone with hints for essay questions for the exam that will be on Tuesday. Please make sure you understand that the exam is not only on the essay questions, it's on all of the material that we covered in all of those chapters. So if I don't see you on Thursday, I hope you get some nice rest. I know you'll be very upset if we don't have class on Thursday. And be careful in this crazy snow that we're having. And I hope to see you all, if I don't see you on Thursday, on Tuesday of next week. Remember, if you are not there to take your exam, you better be sick enough to be giving me a doctor's note so you can make up the exam. Have a great weekend. See you soon.